Welcome friends, we will start with Mycology Part 2. So in Part 2, we will study about fungal growth. Okay, so as we see the growth in different organisms, it will be kind of similar in case of fungus. So let's begin. Fine. So when there is a growth, there is an irreversible increase in the volume, okay, as it happens in the growth. And the mycelial fungi, they exhibit extension growth of the hyphae. Okay, the hyphae will extend, the there will be extension of the hyphae, and there will be increase in the biomass. And unicellular fungi, which are yeast, they can exhibit increase in individual cell volume and increase in biomass. Okay, similar. So the number of yeast cell within a culture may also increase, resulting in the increase in biomass of the culture. Okay, and fungal colonies, they show a differentiation from the margin to the center of the colony. The differentiation is from the margin to the center. Okay, and in case of yeast, the number of cells can also increase. So first, the extension zone. What is extension zone? That hyphae are advancing into fresh media, and branching patterns tend to be at 90-degree angle and monopodial. Okay, monopodial, what does the term monopodial means? That the tip part is growing actively. Okay, and it's elongating. So branching is at 90-degree angle, and the branches, they are arranged to give rise most effective colonization and helps in utilization of the substrate. Okay, the growth of the hyphae is in such a way that it does not overlap other hyphae. Okay, and the mechanism how the hyphae grows and how they are branched is unknown, but uh, probably they are related to food gradients and toxin gradients. Okay, they grow in the direction of the toxin gradients or food gradients. So one is extension zone, another is productive zone. So what is productive zone? Here the major increase in the biomass occurs. So first, in the first phase where the hyphae extends, second phase where the biomass increases, that means more branching, more aggregation, mycelium is formed, and the hyphae becomes thickened. And third is fruiting zone, where biomass gain has been ceased, but the spores are formed. Okay, which is equivalent to the stationary phase of the batch culture, means no more growth will occur. Okay, aging zone is what? Aging zone is what? That vacuolate or empty hypha are present. Okay, autolysis will occur, self degradation. This is equivalent to decline phase of the batch culture. So aging zone is the decline phase, fruiting zone is the stationary phase, right? So what is the characteristic of S-shaped growth curve in case of fungi? So rate of growth or cell division is very low. Okay, growth and cell division is very low. The fungals are adapted in such a way to growth conditions. So it is the period where the individual fungi are maturing and not yet able to divide. Okay. So and during this phase, which is the growth or cell division phase of fungal growth cycle, RNA is synthesized, enzymes and other molecules are synthesized, and this phase is not dormant because growth and cell division occurs in this phase, but growth and cell division is very slow. Okay, another is growth or cell division then starts to accelerate. So growth and cell division was slow in the first phase, then they start to accelerate into the exponential phase in the second phase. And for example, unicellular yeast fungi, if one cell is produced, two cells are formed from one cell in a given period of time, and 2 to 4, 4 to 8, 8 to 16, and so on. Okay, so this represents a period when the fungus is growing or multiplying most rapidly, and it depends upon the growth conditions. which affects the frequency of the cell division events and the probability of both daughter cells surviving. At this accelerated phase or exponential phase, this phase will continue until one or more nutrients become limiting. Okay, until nutrient is limiting, this phase will continue, and oxygen until oxygen becomes depleted or some of metabolites reach to the toxic level. Until and unless these are limiting, this phase will continue. After the accelerated exponential phase, there will be the decline phase. Okay, aging phase. So, between this accelerated phase, there is a stationary phase. There is another phase which is the stationary phase. And after the stationary phase, there is the decline phase. Okay, and during the decline phase, there is no discernible change in cell concentration or biomass, and what happens in the decline phase, what they have done, they have divided this phase into two: stationary and decline. Okay, when there is no changes in the growth, no changes in cell concentration, no changes in biomass, then it is a decline phase. Sorry, then it is a stationary phase. Okay, and when there is a autolysis, it is equivalent to the death phase. Okay, and finally cell death and lysis, it results in decrease in cell number or biomass where observed. When there is stationary, there is no increase but neither decrease. But when there is decrease, it is the decline phase. So how the growth curve looks like: first is lag phase where there is very slow growth, then the exponential phase where is the fast growth occurs, 2 to 4, 4 to 8, 8 to 16. Then I think there should be stationary phase first, then decline phase. Stationary phase is when there is no increase neither decrease. Okay, this is the stationary phase. And then the decline phase when it starts to decline, and then finally autolysis. Now let's start with fungal sporulation. So fungi, they are capable of reproducing both sexually and asexually, and they are able to produce more than one type of spore. Okay, and each spore have the role in their life cycle, and they play an important role in determining whether a fungus forms, and the environment will play a very important role to determine whether sexual or asexual spores are produced. And spores, they are formed as a fungus depletes its energy sources. So when a fungal depletes its energy sources, they form spores. That is in kind of adverse situations. And there are several environmental stimuli or triggers which may be involved in like deciding which kind of spores will be formed. And there are fungi also which are unable to form spores, okay, under high nutrients also, and they are able to grow vegetatively in abundant nutrient conditions. Okay, so the example is wood rotting fungi, which require inoculation and growth on wood blocks before basidiums formed in the laboratory. Okay, so a fungi named wood rotting fungi, what they do, they require inoculation and growth on wood blocks before basidiums formed in the laboratory. Okay, there are others. For example, the exhaustion of a key nutrient, they induces sporulation. When the key nutrient is not there, sporulation is formed. There are another examples: low carbon or nitrogen ratio induces the formation of hyphal arthrospores. Okay, low C by N ratio, low C by N ratio forms arthrospores, and depletion of carbon regardless of nitrogen status induces the formation of conidia. So when nitrogen is like it's normal, but carbon sources is depleted, carbon compounds are depleted, in that case conidia are formed. In case of fusarium, it occurs. In some cases, temperature also influences the formation of spores and it forms sporocarps. And sporulation takes place in the narrow range of the temperature than vegetative growth. Okay, vegetative growth occurs in a wide range of temperature. And the sporulation also require some, in some cases, aerobic conditions. In some cases, fungi light initiates the sexual structures. Interesting, right? And in another cases, pleospora fungus of plant surface, UV induces ascocarp formation. UV induces ascocarp formation. We will study one by one in detail. And also when we will be studying several genus of the fungus, we will be discussing all of these. And light needed for basidiocarp initiation in Schizophyllum, a wood degrading fungus. Okay, so in some cases light can form ascocarp, in some cases light can form basidiocarp, and light and night needed to complete the formation of sporangia of photobolus, a dung fungus. So these are very important examples you need to learn and also remember. Uh, and we will already be discussing in the future videos about all of these. So in some cases, light is required for ascocarp formation, in some cases, basidiocarp formation. In some cases, light and night both are needed for sporangia formation like pylobolus. Now, what is the purpose of sporulation? Why the sporulation is performed? So, first of all, dispersal, and second, preservation, and third, genetic variation. So, these spores which are formed, they are what? They are actually a portion of fungus protoplasm, okay, which are dispersed away from the site where the organism is located currently. And dispersal spores are what? Zoospores, sporangiospores, and conidia. Sporangio, and conidia. Another is for preservation. There are spores which preserve the potential for growth. Okay, they helps the fungus to survive under unfavorable conditions. These are oospores, zygospores, and chlamydiospores. They are preservation spores which do not grow, but in the adverse situation they just become dormant in the form of spores. And genetic variation, where sexual sporulation provides the potential for genetic variation. Okay, and there are several factors which influences the type of sporulation: the genotype of the species, genotype of mycelium, and the extent of somatic growth and environmental condition. So genotype of the species means there are several species which has own genetic, like every species have their own genetic program. Genotype of mycelium is where one strain behave different from another, that one mycelium different, one hyphal strain uh, differs from another. Extent of somatic growth, a certain amount of growth is often necessary before sporulation occurs. Okay, and like there are several conditions which influences or stimulates the formation of spores. It is temperature, light, and specific nutrients. So what are the general characteristics of fungal spores? So spores represent they are microscopic in nature. They can be dispersal, or they can help in preservation or survival propagules. They are produced by many or most species of fungi. They are variable in size, shape, and color. They can be unicellular or multicellular. Conidia, which are produced by alternaria, are multicellular. There are some spores which possess a textured or ornamented surface, like surface have some kind of sculptures. An example, uridospores of Melum spora epita. It has some kind of sculpture. It is a causal organism of willow rust. There are the protoplasm of spores. Most of the spores, it is surrounded by a rigid wall which is thicker and more multi-layered than that of somatic cells of the hyphae. Okay, and there can be pigments also in it, melanin and lipids. Okay, so some protoplasm of the spores, some of the spores protoplasm is covered by the rigid wall, which may have some pigments and lipids, and they are thicker than the somatic cells. So spores contain nutrient reserves, which may be in the form of lipid, tree halos, and glycogen. Spores are having low water content so that they can become dormant, and they have very low rate of metabolic activity. They vary in primary function, which may include dispersal to a fresh site or host. Okay, survival at the same site and increasing genetic variation. So they also vary in the methods by which they are formed, released, and dispersed. So these are general idea about the fungal spores. So next we will start with individual spores, how they are formed, what is the procedure, in which organism they are formed, what is the method, sequence, and all. So next we come to method of conidiogenesis. So the conidium formation or production, it involves the transformation of a part of a cell into separable spore. Okay, part of a cell forms a spore, and the cell, the hyphae which are there, they are separated into septa, okay, by segmented cells known as septa, and these separate cells they then thicken and swell and finally separate. Okay, the hyphae form septum, and this separated cells they become thicker and they finally separate. And this kind of conidiogenesis, where the septa appear before the conidium initiation occurs, it is called thallic. Okay, it is called as thallic. When the new conidium is initiated, it begins to swell or thicken, and then it is cut off by a septum. The conidiogenesis is called blastic. Okay, and when the cutoff is later on, it is blastic. Okay, so when septum forms first, then it is thallic, and when septum forms later on, it is blastic. Okay, so let's see the picture. So this is the one where the septum will form. Just a moment. So see here, where the septa appear before the conidium initiates, it's called thallic. So let's see figure A and C. So here the septa, it is showing that here it's the septa which is forming. Here the septa which has formed before. Okay, the septa which has formed before, the cells then become thickened. Septa cells then become thickened, and they are individual spores which then separate. Okay, this is thallic. And what about blastic, where the septa forms later on? Okay, there is no septa yet formed. Septa forms later on. First, there is the thickening and then the septa formation. See here, new conidium is initiated. It begins to swell or thicken. New conidium is initiated, begins to thicken. Septa forms later. This is what? This is blastic. So there is another one. Septa forms later. This is also blastic. First thickening. Here also you can see this picture number A is not very clear, but it is holothallic. Multiple cornidia are produced simultaneously. This is holothallic. Okay. Second is holoblastic. Third is enterothalic, and fourth is enteroblastic. Okay, so when multiple conidia are formed simultaneously, it is holoblastic. When again in or holothalic, and when the multiple conidia are produced uh, kind of in series. Okay, I will discuss about it gradually. So see here, when the wall of the conidium is continuous with the cell that produced it, it is called either holothalic when thalic or holoblastic when it is blastic. And in another case, when only the inner walls of the conidium bearing cell are involved in conidiogenesis, then it is entero is used. So you can see that here the inner wall is involved in the formation of either thalic or blastic spores. It is known as entero, that means inner. Okay, so enterothalic, their types are very rare. Enterothalic or enterothalic are very rare, but enteroblastic types are the most common ones of all. Okay, so see here enteroblastic once again, where the inner wall first of all participates in the formation of the spore, and this is the most common type of formation of conidiospores. So what is the sequence? If only a single conidium produced on conidiogenous cell, which is uh, which is here, if a single conidium produced on a conidiogenous cell, we can hardly speak of a sequence, how it is formed. But when more than one conidiogenous cell is produced, they can occur simultaneously. Okay, they can occur simultaneously. I'll show you its picture. When a single conidium is formed, the sequence is hard to determine. Okay, but in case where multiple conidia are formed simultaneously or serially one after the another, we can tell about the sequence of the formation. So say for example, here it is, this is a single conidiogenous cell, it is holoblastic in nature. Okay, holoblastic means what? Holoblastic means what do you what did you understand about holoblastic and holothalic? Holoblastic was when the septa forms first and then when the septa forms later, first there is thickening, and when septa forms first, okay, when septa forms first and then the thickening occurs, it is thallic. Okay, so here you can in the sequence we can say whether it is a progressive at the retrogressive locus or a progressive locus. Okay, in the B case you can see that it is retrogressive. Multiple conidia are formed from a retrogressive locus. And okay, and you can see that retrogressive means it is forming, the new ones are forming below. Okay, and holoblastic, you can see here multiple conidia are produced in a progressive locus. Progressive, see here, first this is formed and then this and then this, so it's one is forming above the another at the progressive locus, the locus next to it, and in the retrogressive, the locus downward to it, the spores are formed. So what are the types of asexual spores? It can be sporangiospores or conidia. Conidia we already discussed. Let's understand about sporangiospores. So sporangiospores, they are endogenous in nature. They are formed within the sporangium. And how they are formed? They are formed because of the cleavage of protoplasm around nuclei. Okay, protoplasm is cleaved around the nuclei. There is a wall around each nucleus of the portion in some cases, and these kind of fungi belongs to Chytridiomycota, Oomycota, Hypochytridiomycota. And there are two, they include two types. Asexual spores here, sporangiospores include two types. What they are? Zoospores, which are motile in nature, and aplanospores, which are non-motile in nature. So sporangiophores, they are within the sporangium. They are formed by the cleavage of protoplasm, and they form a wall around the nucleus. They belong to Chytridiomycota, Oomycota, Chytridiomycota, and there are two types: zoospores, which are motile, and aplanospores, which are non-motile. Now, what are conidia? Conidia are exogenous cells. Okay, they are formed at the tip of the supporting hyphae called conidiophores. Okay, so conidia are formed at the tip of the supporting hyphae, and this is known as conidiophore onto which they are formed, and they are developed in variety of ways. As we have discussed earlier, that was just the basic about the conidia. Let's study about the conidia in detail. So they are developed in variety of ways. Conidia, the characteristics include mitosporic fungi and the fungi belonging to Asco and Basidiomycota. So these conidia includes two types, thallic and blastic. And don't worry about the examples, we will study about it in the subsequent videos. So thallic conidia are what? These are origination from septation and then fragmentation of the hyphae. So first septation occurs and then the wall thickens, as you remember, and they may develop in intercalary or central position of the hyphae. So it can be at the central position, it can be at the tip of the hyphae. All the layers of hyphal wall, they are involved in spore formation. So you can see here that this is a conidiophore which bears conidia, conidiophore, and these conidia can be single in number or multiple, and there is sterigmata just below the conidial spores, which is the bulged cell, and below the sterigmata is medullae, and the larger one is vesicle at the mid in some cases. Okay, so blastic, they are characterized by developed by budding or swelling process. So blastic conidia, they are generally formed by budding or swelling, but thallic are generally from septation. Okay, so in this case also they can be single or multiple in succession to form a chain of spores, and these blastospores, they are characterized by forming a budding of the hyphae. You can see that the budding is being formed. In case of yeast, you have seen that, and here both wall layers are involved. The spores are attached and the bud further blastospores, and this gives rise to branched chain of spores. The blastic have two types. One is blastospores and another is sporospores. Okay, there are two mechanisms of forming blastic conidia. One is blastospore where budding is seen, and another is sporospores where the scar is formed at the point of detachment. What happens in case of porospores? Porospores, so blastic, since blastic is more common types of conidia formation, there are several types of blastic conidia formation. One is blastospore, another is porospore, third is aluriospores, fourth is alenospores, fifth is phyllospores, sixth is arthrospores, seventh is chlamydospores. Next is zoospores. Then comes the sexual spores, which we will cover in the next video. So what about porospores? So the developing spore here emerges through a distinct pore in the hyphal wall. Okay, there is a pole in the hyphal wall through which these spores emerges, and the inner layer of the hyphal wall, it is involved in spore development. Okay, so in case of blastospores, both wall layers are involved. Here only inner wall is involved. Okay, and the new spore which then develops its own new layer wall. Okay, the new spore which forms, it develops its own inner layer. Okay, this is the inner layer, and here it forms its own inner layer. Okay, so the outer spore wall is often thickened and pigmented. Maybe we have studied melanin, and a scar is produced once they are separated. So scar is produced at the pore region at the point of detachment. And this is what? This is conidiophore. Next is allureospores, where they are developed as a single and terminal spores. So single, only one, and terminal spores, and they are separated. So they first inflates, they become inflated or like bulges, and become separated by a septum at a very early age in spore development. Septum occurs earlier here, not like thallic, but kind of a little earlier. And here again, both wall layers are involved. So we studied only one wall layer is involved in porospores. In allureospores, both wall layers are involved, and the spores have wide truncated scar. So scar was there in case of porospore also, but here wide scar, wide and truncated scar. Okay, so no further development will occur at the point of detachment from where the spore will detach. So the next spore usually has to develop by production of branch below the scar on the conidiophore. So next spore is here on the branches. Okay, this is allureospore. Next is anellospore. Anellospore is where conidia forms in such a manner which is similar to allureospore. Here a new growing point does not develop at the scar. Okay, so there we found that a new growing point develops at the side of the scar, but here no new growing point will develop at the scar region, and then a chain of spores may develop. Anellospores is known for the chain of spores which develop, and here the conidia gets a little longer when each spore is formed. Okay, and annellations, which is ring like scar, is observed around this elongating portion. You can see the ring like scars are formed, and it forms chain of spores. So each annulation or each ring will represent the production of one annulospore. So the number of spores it is producing in the chain, that number of rings will be formed. Okay. So next is phyllospores. Here again the spores are formed in succession, and each spore pushes up the tip of the conidiophore. Okay, which is called now as a phyllide. Okay, each spores it is pushing up the conidiophore, which is known as firelight. This is firelight. It is pushing up the conidiophore, and the spore wall is new and distinct from both layers of the firelight. The spore walls which are formed, they are distinct from both the layers. So this layer 1 is this, layer 2 is this, but this is very different. This wall is different from the conidiophore. And the first spore has a cap like structure. First spore has a cap which represents the tip of the phyllide wall. So what is phyllide here? Phyllide here is when one spore is formed, it pushes the tip of the conidiophore. So when a spore is formed here, when a conidiophore bearing the spore at the tip, and when the tip part pushes the conidiophore, it is known as phyllide. Okay, and the tip part here have the cap which through which the spore emerge. Okay, and all other spores in the chain are smoothly and rounded. So all other spores are smoothly and rounded. So tip of firelight, it's visible on first spore. So you can see that if this is a conidiophore, and when the another spore like structure is formed, it has pushed, you can see the wall of conidiophore and the cap is same. So this spore has been formed by pushing the phyllite. Okay, by pushing the phyllite, this spore is formed because you can see that the two layers of the cap and the phyllite or conidiophore is same. Okay, so this is where the next spore which is formed is smooth, and they are formed in chain. Next is arthrospores. Arthrospores is characterized by septation and fragmentation of the existing hypha. There is the septation and the fragmentation, and each fragment and the elements of the hypha, it becomes converted into conidia. Okay, and each fragment will round off. So remember, septation and fragmentation both occurs in arthrospores, and each fragmentation rounds off in leverate succession. It forms a kind of cells. Okay, here it's just a septation, here it is rounding off, and they become separated from one another due to breakdown of the middle region. This is arthrospore. Next is chlamydiospore, very common and important. Here, chlamydiospores are a type of resting or survival spore. Okay, it is a survival spore. They are found in several groups of fungi, and here the intercalary cell or hyphal cell or compartment enlarges. So remember, intercalary cell enlarges and rounds up and develops a thickened, often pigmented wall. Okay, they have dense cytoplasm, more nutrients, and the layers, all the layers are involved in spore formation. So remember, only one layer we generally saw in porous, and uh, means altogether new layer of the spore is seen in phyllite, and they developed under the condition of stress conditions that are unfavorable for normal somatic growth. Okay, under stressed condition, these kind of survival spores are formed where intercalary cells takes part in the spore formation. And the last one is zoospores, which are motile sporangiospores, in where the sporangia in which they are formed are called on which they are formed are called zoosporangia. Okay, there is no wall which surrounds the zoospores. They resemble flagellate protozoa because they are motile in nature, and the fungi that produce them will require some water at some stage during their life cycle. So water is required for zoospores, and there are three different types of zoospores. What are they? Chytridiomycota, Hypochytridiomycota, and Oomycota. So three different types of zoospores are there which distinguish these three types of species: Chytridio, Hypochytridio, and Oomycota. So in case of Chytridiomycota, there is there the zoospores have one whiplash structure. In case of single posterior smooth whiplash flagellum. In case of hypochytridiomycota, they have one anterior, okay, they have one anterior tinsel flagellum, okay, single anterior tinsel flagellum, and in the third case, Oomycota, they are biflagellate, where one is whiplash and one is tinsel. Okay, so these were all about the sexual spores of the fungus. In the next class we will study about the these are all about asexual spores. In the next class we will study about the sexual spores. So what we studied today, let's recall once. So we started with fungal growth, the different phases of fungal growth: extension, productive, fruiting zone, aging zone. Characteristics of S-shaped growth curve: it's like first there is lag phase and exponential phase, then in the most cases stationary phase. White has shown decline phase, don't know, stationary decline, and then lysis phase. And then the fungal sporulation. So there are several types of spores which are formed under adverse conditions. So the purpose of sporulation is either dispersal or preservation. Dispersal spores are zoospores, sporangiospores, and conidia, and preservation are oospores, zygospores, chlamydiospores. We'll study one of them in detail in subsequent videos. And there are factors affecting the type of sporulation, which is genotype of species, genotype of mycelium, extent of somatic growth. Uh, like certain amount of somatic growth is necessary, and then a specific environmental condition which triggers spore formation. Then general characteristics of fungal spores, method of conidiogenesis: blastic and thallic. Blastic, where septum form first then thickening occurs, and in thallic septum form first and then thickening occurs, but in blastic thickening occurs first then the septum forms. And there are different types of asexual spores. We studied about conidia, and then we studied about the thallic conidia and blastic conidia. Blastic conidia are characterized by several, like blastospore, porospore. Okay, uh, porospore having only formed from only inner layer of hyphal wall. Then aleurospore, where the single terminal spore is formed, and then second spore is formed from the next spore below the truncated region. Alenospore is where the spores are formed in chain and annihilation ring like scars is formed. And phyllospores, where the conidiophore is pushed up to form the spores, and here again chain of spores are formed. Arthrospores, where septation and then fragmentation occurs. Chlamydospores, where intercalary cell becomes enlarged, thickened, and forms the spore. Zoospores, which are motile in nature. There are three types, which distinguishes three kind of species. Chytridiomycota, having only single flagellum from the posterior side, and Hypochytridiomycota, having single flagellum from the anterior side, but it's tinsel flagellum. And in case of Oomycota, they are biflagellated, having one whiplash and one tinsel flagellum. So we will study about the sexual spores in the next video. Thank you so much.